Good day. This is the Professor, Professor CBMD PhD. And today's Thursday, November 3rd, 2022. It is 1:58 and 46 seconds. AM Eastern Standard Time. And today's research, cancer cells. So let's hop to it here. The cancer cells, exposed to high viscosity, move better, and their metastatic potential increases. Date November 2nd. 2022. Source: University of Barcelona. An international team of scientists has discovered how cancer cells exposed to high viscosity environments change the way they move to improve their invasiveness and favor metastasis. The studies provide new targets for the design of possible cancer therapies. Understanding how cancer cells move and make decisions in these confined environments is important, as 90% of cancer-related deaths involve metastasis. Researchers have been working together over the past 6 years to unravel how cancer cells use ion movement to mechanically activated ion channels stimuli that deform cell membranes in order to adapt their movement to different mechanical stresses and environments. In these two new studies, the scientists asked themselves how cancer cells polarize ion transport mechanisms in the leading edge and trailing edge of the cells to move through narrow spaces, and how cancer cells optimize movement when fluid viscosity is high. And to address these important questions, they studied the movement of cells in three-dimensional media generated using bioengineering techniques which resemble the pathways along which cells normally move in our bodies. The key proteins were isolated within the cell using high-resolution microscopy. Cell volume, ion movements, and electrical activity were recorded, and they evaluated how the expression of different genes that are important for the progression of cancer changes. The research team found that cancer cells can move in confined spaces by simply taking in water at the leading edge of the cell and releasing it at the trailing edge. And they do so without the need to establish molecular interactions with the walls of the surrounding tissue. In real life, this is possible because at their leading edge, the cells accumulate an ion transport system. The sodium proton exchanger at AG1, which charges the cell with sodium, which increases osmotic pressure and favors the entry of water into the cell. At the same time, cancer cells concentrate the SWEL1 protein in their trailing edge. SWEL1, also known as LRRC8A, a chloride channel activated by increases in cellular water content that facilitates the exit of chloride and water. The end result of the coordinated action of these two ion transport systems on the leading and trailing edges enables cell movement. More importantly, the study shows that the activity of these two systems is essential for the movement of cancer cells outside of blood vessels and the development of metastasis. In the second study, the scientists questioned how changes in viscosity in the cellular environment can condition the way cancer cells move and behave. The viscosity measures the resistance that a fluid exerts on anything that moves in or with it. As such, common sense and fundamental engineering indicate that inert particles move more slowly in high viscosity media. The scientists have now demonstrated an effect that a priori may seem counterintuitive. High viscosity promotes the migration, invasion, as well as the extravasation of tumor cells, exciting from blood vessels and lung colonization. The cells of our body are constantly exposed to fluids of varying viscosities in some pathological situations, such as tumor growth. The local viscosity surrounding the initial tumor increases due to abnormal protein degradation or compression of the normal drainage pathways the lymphatic vessels. In addition, the cancer spreads to other parts of the body. Cells have to travel through spaces filled with interstitial fluids and blood, which are more viscous than water. In previous studies, the team demonstrated that cells adapt to high viscosity situations by activating a protein called drpv4, an ion channel that facilitates the entry of calcium into the cell, otherwise impossible due to the lipid membrane that delimits the cell and is impermeable to ions. Calcium is an element that, when increased inside the cell, controls various cell functions. The research team posited that cancer cells exposed to high viscosity may use a similar mechanism to enhance their motility and dissemination, and they were right, but with interesting surprises. And by exposing cancer cells to high viscosity, they observed that the first cellular element that responded to the stimulus was a protein actin, which is part of the cytoskeleton and shapes the body of the cell. And this initiates a cascade of molecular events that ends with the activation of the TRPV4 channel, which in turn activates a cascade of intracellular events that result in the reinforcement of the cell cytoskeleton and the activation of motor proteins. Interestingly, by means of all these changes, the cells modify the means of migration and no longer employ the movement of water. In these conditions, they use their cellular skeleton and muscles, as well as interactions with surrounding walls, to propel themselves faster. It is as if the cells had gone to the gym to train hard under high viscous loads and perform better when they are physically challenged on their journey from the primary tumor to their final destination in distant metastasis. The study also found that cells not only move faster when surrounded by high viscosity fluids, but also when they have been previously exposed to such fluids and then removed. In other words, cells can not only detect and respond to elevated viscosity, but can also develop a memory of their exposure to this condition. The vast majority of cell biology research is conducted in cell culture media with viscosities close to that of water. Researchers defined for the first time how cells detect and respond to the physiologically relevant levels of fluid viscosity in which they are commonly found in the body of healthy and sick patients. The definition of the molecular mechanism used by cells to adapt to changes in viscosity of the media was a 2D force in which they had to change their perceived idea in which cellular elements are the first to respond to this type of mechanical stimulus. The great coordination between the structural elements of the cells, their actin and myosin cytoskeleton, with the mechanisms of ion transport and water that regulate cell volume marks a major breakthrough in our understanding of cell mechanobiology. It will be very informative to examine how primary tumors and cancer cells that spread from primary tumors respond to local changes in the viscosity of extracellular fluid found in the body during disease progression and during invasion into the tissue microenvironment. The development and optimization of biosensors that allow real-time measurement of extracellular fluid viscosity, along with imaging of cancer cells and live animals, will be crucial to address this point. At this stage, we cannot propose a specific molecular intervention to combat cancer metastasis. Researchers believe that the molecules and pathway they've identified in the study can be used as pharmacological targets for possible cancer therapies. And this is the professor of microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.